Gene transfer methods in animals' embryonic stem cell. Gene transfer using embryonic stem cells, ESCs, is a powerful method for introducing genetic modifications into animals. Embryonic stem cells are pluripotent cells derived from the inner cell mass of early-stage embryos. They have the unique ability to differentiate into cells of all three germ layers, making them valuable for gene transfer and the generation of genetically modified organisms. Overview of gene transfer using embryonic stem cells, ESCs. Isolation of ESCs. ESCs are isolated from the inner cell mass of blastocyst stage embryos, 3.5 to 4.5 days post-fertilization in mice. Culture and expansion. ESCs are cultured in vitro with specialized media and feeder cells to maintain their pluripotency. Introduction of transgenes. Genetic material is introduced into ESCs via methods like viral transduction or electroporation. Selection and cloning. Cells with the transgene are selected using markers, e.g., antibiotic resistance. Cloning techniques isolate ESC lines with the desired modifications. Verification. Genetic modifications are confirmed through PCR, Southern blotting, or sequencing. Differentiation. Modified ESCs are induced to differentiate into specific cell types by adjusting culture conditions. Injection into blastocysts. Genetically modified ESCs are injected into blastocysts, which are implanted into a surrogate mother. Germline transmission. Surrogate mothers carry embryos to term, resulting in chimeric animals with the genetic modifications in their germline. Breeding transgenic lines. Chimeric animals are bred to establish stable transgenic lines with the desired genotype. Types of embryonic stem cells, ESCs. Embryonic stem cells, ESCs, are pluripotent cells from the inner cell mass of blastocyst stage embryos, capable of differentiating into all three germ layers. They are crucial in regenerative medicine, developmental biology, and genetic research. The two primary types are human embryonic stem cells, HESCs. Derived from the inner cell mass of human blastocysts, typically from surplus IVF embryos. Can differentiate into a wide range of cell types. Valuable for regenerative medicine, drug discovery, and understanding human development. Ethical concerns exist due to the destruction of human embryos. Mouse embryonic stem cells, MESCs. Derived from the inner cell mass of mouse embryos. First ESCs to be isolated and characterized. Extensively used to study gene function, development, and to create transgenic mouse models. Crucial for insights into mammalian biology. Applications of embryonic stem cells, ESCs. Embryonic stem cells, ESCs, have broad applications due to their ability to differentiate into various cell types. Key applications include regenerative medicine. Differentiating ESCs to replace or repair damaged tissues and organs, potentially treating conditions like heart disease, diabetes, and neurodegenerative disorders. Drug discovery and development. Using ESCs to create specific cell types for disease modeling, aiding in studying new drugs' effects and streamlining drug development. Understanding developmental biology. Investigating embryonic development processes by manipulating ESCs to study cell differentiation, tissue formation, and organ development. Modeling diseases. Creating in vitro models of diseases by differentiating ESCs into affected cell types, aiding in the study of disease mechanisms and development of treatments. Creating transgenic animals. Injecting modified ESCs into embryos to produce chimeric animals with specific genetic modifications, used to study gene function and disease models. Cell replacement therapies. Using ESCs to generate cells for replacement therapies, such as insulin-producing cells for diabetes or neurons for neurodegenerative disorders. Understanding stem cell biology. Studying how ESCs maintain pluripotency and differentiate, providing insights into stem cell biology. Tissue engineering. Creating artificial tissues and organs by guiding ESC differentiation, aimed at generating functional tissues for transplantation or in vitro study. Advantages of embryonic stem cells, ESCs. Pluripotency. ESCs can differentiate into all body cell types, making them highly versatile. Tissue regeneration. They can be used to generate specific cell types for replacing damaged or diseased tissues in regenerative medicine. Disease modeling. ESCs can create in vitro models of diseases, aiding in studying pathogenesis and testing therapies. Basic research. They are a key resource for studying embryonic development, tissue differentiation, and cell biology. Transgenic animal generation. ESCs facilitate the creation of transgenic animals, enabling the study of gene function and disease models. Disadvantages and challenges of embryonic stem cells, ESCs. Ethical concerns. Derivation involves destroying human embryos, raising ethical issues. Tumorigenicity risk. Undifferentiated ESCs can form tumors if injected into the body. Immunocompatibility issues. Risk of immune rejection during transplantation. Limited availability. Requires surplus embryos from IVF, leading to limited and controversial sources. Genetic and epigenetic stability. Potential for changes during culture affecting stability and functionality. Complex differentiation protocols. Challenges in efficiently directing differentiation into specific cell types. Developmental stage specificity. Limited in modeling diseases manifesting later in development. Alternative technologies. Ethical and practical challenges have spurred development of alternatives like induced pluripotent stem cells, iPSCs. Thank you.